Welcome to the next step. The second step is to prepare the topological files. So we need to prepare the protein topology file, and then we need to prepare the ligand topology file by using the different tools. For the protein topology, we will use a command which is native to the GROMACS. We will use it and we will prepare it. For ligand topology, we will use different tools. So let's split it into the part 2A, in which we prepare the protein, and then there will be the part 2B, in which we prepare the ligand topological file. So for protein topological file, here are certain options that we need to use a force field. Okay, force field in this tutorial particularly, you can use the charm 36 force field. So there are a lot of versions are available in the charm force field. You need to open this website, and here you can see that we got different charms force field available. We got here February 2026 force field. We got here the charm force field, which is 5.0 version, 4.6 version, and we got here the older ones. So why not we just download this new version. Actually, I will just save the link as, and I will just put it into my this particular folder, and I will save it. So it has been saved as such. You can also see other actually this force field. If you really need the old one and you need to modify when you are advanced user, you can also modify the force field, but we don't need it for now. So the other thing which we need from this website, actually, that they are the scripts. So these scripts we will be in requirement, which are required to prepare the ligand, actually the ligand bonding and other things. And for that purpose, I will just download this script, which is third one, NX2, which require Python 3 version and networks 2.3. So again, I will right click on it. I will say save the link as, and I will destinate it to our folder, and I will save it. So this is actually the Python file which we have saved it. So we don't need anything, but if you want to read about the force field, the website is open for you. You can dig it into more detail. So we are back here. So we have downloaded force field, which is not from 2022, but which is from 2026. So what we need now we need to use this GROMACS commands, actually, that how these things they work. So for that purpose, if you remember that my folder is on desktop, okay, it's not present in the Linux area. I will copy it. So why not I just copy it and I paste it into my Linux here somewhere in home folder and somewhere where I was working. Okay, and here I have pasted it. That is ligand underscore T. Now let me open the Ubuntu here. So I have opened the Ubuntu here. Here I will go to, actually, why not we do the ls. So here are certain things which are present here. You can see we got the ligand underscore T. So I will choose cd ligand underscore T here, and here you can see that we got these number of files which are present here. We got the ligand and we got the proteins. So we got these things actually here. So first our command means, first thing we need to prepare the topology thing, that is to use gmx. Gmx is native command actually. When you type it, that means you are using GROMACS. And then you will use, okay, let me type the whole command actually here, copy and paste, and then I will explain it to you. So our protein, that name is protein.pdb. If you remember here, we named it as protein.pdb. So what we need to do, pdb to gmx, and then we got here the file which is protein.pdb, and this is the output what we need. Output name I will also change. I will just give it a name which is protein underscore processed. I will just say it protein.gro file because we just need only one grow file here, and then here we will do ter. Okay, so this is our command. Let me just confirm it again. You can also confirm it, you can just copy and paste. So this is ter. Okay, you can copy and paste, you can use their names or you can use your own names for the understanding. And this command that will write the topology for our protein file. So let me just enter it, and here it has started working. Okay, it says GMX is not found. What does it mean? It means Chromax is not found. If you remember that last time in WSL, we got some. So we countered these errors many times. That Chromax is not found, you need to install. We don't need to install it actually. Sometimes on WSL, we get these errors. We need to source GMX and we need to activate it. So let me check GMX and then I will say it version. So it shouldn't display anything. Okay. Then what you need to do, you will go to this command which I gave you many times, or you can write it down. Source user local chromax bin and gmxrc that we used into our tutorial when we were doing the chromax installation actually. And here you will just paste it and you will enter it. Now let's check gmx version. Now you can see we got the chromax version which is 2025. Okay, let's repeat the command here which we made it here, and I will press enter. So for the topological file, just to need to understand, it's saying that select the force field which is present here. Okay, you need this particular force field which is present amber 03 something. You need this force field, or what kind of force field actually you require. You require the charm force field, or you don't require the charm force field actually. What kind of force field you require. So there are a lot of options, but here issue is that the charm force field which I had downloaded, it's not showing here. What's the reason? Reason is that we didn't unzip it. So we need to unzip this particular thing. I will just go here and I will say it, unzip it for me. Okay. What it will do, it will unzip it for me, and then that's force field that will be available. So here you can see that we have unzip it, and in WSL there are a lot of problems. It refreshes like this. Okay, we got here .ff or the force field file here. So let me do here, let me again run it. Okay, control C, I will stopping it. Let me clear it. Again that command I am repeating. I will press enter, and here you can see that either this particular force field which we are gonna use, that is available or not. That was the charm force field which is present here. So this charm field force field we are gonna use, but again we got an issue that force field didn't appear. Reason is that I just do the things, I just unzip it like this, that force field folder contain another force field folder. So let me remove the first folder. Let me just cut and paste because it is not reading the force field. So it should be as a major folder, and force field file that should be present here. So let me control C again. Let me do a clear thing and let's run it. Now here you can see it says from the current directory charm force field or atom force field. This is our 2026 force field. So you can get these lot of errors actually. So what you need to do, I'm showing you that particular things, that if you unzip it like this, where the files will go, you need a major folder, and so you need to cover it up. So one option I will select, I will say one, I will press enter. Now it's saying which model. Actually it's giving you option of selecting this model, of selecting other models. I will use the one. I will press enter, and here you can see it's preparing the topological file and asking you on the N terminal what should be present. We know N terminal we got NH3. We don't need methionine and other things. We need NH3. I will press 1. Here you can see you need COO, COOH. We need in ionized form COO minus. I will press 0. I will press enter. Here you can see that it has successfully generated the topology file from the protein.pdb. So this is our part 2a, that how we can prepare the protein topological file. Let's check that either topology file is present here or not. Here you can see protein.gro is formed, the pole.top is formed, and then we got here position restraint file, posre file.itp. Let's see what's in topological file, because topology file is very important. So let's do it in code, and I will say topology.top, and then it will be opened into our this particular program, which is code. Okay, let me just clean it up so we can see the file here. So this is topological top file here, and this is how the file is generated, and it contains these particular things, the charges and other things, what kind of charges here. And if you remember that there is no charge on methionine, but here it's showing +1 charge. What's the reason? Because this is NH3 which is present here, that's why it's showing +1 charge. Otherwise there is no charge on the methionine side chain. So as in the end, if you go, there are a lot of things how these are connected, which atom is connected to what, and these are our last residues somewhere here, I think 62, and that is the asparagine. COO is at the end, so it should be -1. So this is how we check the topology file. We got the topology file here. And then next, if I wanna check actually the protein.gro file, so let me show you that it's different from PDB. So it's look like this. Its numbering is totally different. Its residues are different. So it look like this. It will just show you that how many atoms and other things are present. So here we got a total of 2614 atoms. So that is important actually, because we are going to combine these atoms into a complex. We are just doing these atoms plus the ligand atoms, and then we will prepare the complex. So this is for now, this is our part 2a, in which we have prepared these files. We got prepared the position restraint file, we got here the topology file, and we got here the protein file, which is made with this particular topological file. Okay, Ji. Our next part will be the preparation of ligand topology. That will be 2b.